First of all, I would like to share my screen so that it will be easier to explain the topic. So be ready, students. Keep your textbooks open. Now we will start our lecture. So very good morning to my dear students and teacher. On the behalf of Bren Mumbai Municipal Corporation Online Education English Medium, I am hereby Bhagyashree Jaiswal from Chinchauli MPS. Today I am hereby to take your lecture of class 9 of subject science 2. As we have started our chapter number 16, heredity and variation yesterday. So today we will take the quick recall what we have seen in our previous lecture. Okay. So in the previous lecture, what we have seen, the topics which we have seen are the topics were heredity. Heredity means what? The transfer of characteristics from parents to offspring is called as heredity. Therefore, every living organism has similar characteristics as its parents. Okay. Then we have seen the topic inheritance. What is inheritance? The tendency to transmit the variations from one generation to the next is called as inheritance. Then genetics we have seen. Genetics is what? Genetics is a branch of biology which studies the transfer of characteristics of organisms in the form of genes from one generation to the next. Then we have seen about the inherited traits and expression of traits. Here traits means characteristics, and the specific characteristics seen in any living organisms are due to the genes transmitted from its parents to the offspring, that is DNA. Yes, and we have seen the examples, that is the growth in the height of the plant. Yeah, in this what is depend? It depends upon the growth hormone. If the growth hormone is synthesized more, then the height of a plant will be more. If the efficient enzyme is not prepared in a greater quantity, then the height of the plant will be short. Then we have seen about the chromosomes. So what are chromosomes? The chromosomes are made up of nucleic acids and proteins, and they have hereditary characteristics located on them in the form of DNA molecule, and the number of chromosomes is specific for every species. Then we have seen the different types of chromosomes also. Different 4 types of chromosomes. What are they? They are metacentric, submetacentric, acrocentric, telocentric, and all these types of chromosomes are identified or they are classified on the basis of the position of the centromere. So yesterday only I told you that in our next lecture we will see about the DNA. So my dear students, I want that you should open your textbook and be with me for my period. Okay. Now DNA. Now what is DNA? DNA, what is the full form of DNA? The full form of DNA is deoxyribonucleic acid. What it is? Deoxyribonucleic acid. So the full form of DNA is deoxyribonucleic acid, and a pet name is given, that is master molecule. Reason behind this I will explain in our further part. So what is the pet name of DNA? It's a master molecule. Yes. It's a master molecule. Now, when a name comes, we want to know about the structure also. So this is the physical, or this is the structure of the DNA. Yes, this is a structure of the DNA. It is in a strand form of this helix. Yeah, it is in the string form. Yes or no? So who has discovered this DNA? Now, Frederick Michael. He began working with white blood cells in 1869. Here, the white blood cells are a major component of pus in the infections. What are the white blood cells? White blood cells are a major component of a pus in infections. As a result, Michael collected lot of pus from the bandages at a local hospital. Okay, what he has done? He collected lot of pus from the bandages at a local hospital and added a weak alkaline solution to the white blood cells. When he did, the cells decompose and cause the nuclei to move out of the solution. What happens when he added the weak alkaline solution? What he has seen, that the cells were decomposed and caused that the nuclei were moving from the solution. And this from the nuclei, Michael isolated a substance known as nuclein. What he has done, Michael isolated a substance known as a nuclein, and after chemical analysis, nuclein was later renamed as DNA. Okay, later it is renamed as DNA. So chromosomes are mainly made up of what? DNA. This acid was discovered by whom? By the Swiss biochemist Frederick Mica. In which year? In 1869, while studying white blood cells. Initially, this acid was reported to be only in the nucleus of cells. Hence, it was named as a nucleic acid. But, however, it was later realized that it is present in the other parts of cell too. It was recognized that it is present all over the body in each and every cell. So the molecules of DNA are present in all the organisms. Okay, from virus and bacteria to human beings. See in this slide. So the DNA is present not only in the bacteria, in virus it is seen, in plants it is seen, in animals, and as well as in a human beings. Okay, so this molecules, which molecule? DNA molecule control the, what is the function of it? It controls the functioning, growth and division. Division means what? Reproduction of the cell, and are therefore called as a master molecules. Yes. So DNA controls what? What is the function of DNA? DNA controls the functioning, growth and division. What do you mean by division here? Reproduction of cell, and it also bears hereditary characters and is responsible for genetic transmission. So it is called as master molecule. So it is called as master molecule. Up till here you understand where it is present, how it is. Now, what is the location of DNA? Location of DNA means what? Where it present, where it locates. As someone asked for us, where do you live? We give the location. So in the same case, that how, where we can locate DNA. So we can see DNA in the cell organisms, that is in the cell, there is nucleotide, nucleus, many things are there. So in the nucleus, see here, in the nucleus, chromosomes are present. Yesterday only we have seen it is in the X type structure, and in this X, or in this chromosome, in the Q arm, this DNA is present. Okay, in the chromosome only this DNA is present. Understand? So this is the location of the DNA. Now, but here, see, we have seen the structure of DNA in this way. But when we uncoil these chromosomes, see here, When we uncoil these chromosomes, we can see so much long coils. These are the supercoils. Here, what is present here? Nucleosome. See the coils we have seen here. And this nucleosomes, there are different groups here. And the single red cells, these are called as what? Histones. And from this only, this DNA structures are formed. So DNA is called as what? Double helix structure. So DNA is having double helix structure. Okay. So what we have seen, that DNA is a deoxyribonucleic acid. Chromosomes are mainly made up of DNA and proteins, and it was discovered by Swiss biochemist Friedrich Meiker in 1869 while studying white blood cells. It is present in nucleus, so it is called as nucleic acids, present in all organisms from viruses and bacteria to human beings, and it controls the functioning, growth and division, that is reproduction of the cells, and are therefore called as master molecules. Yes or no? So they are called as master molecules. Now, we have seen just now the structure of DNA. But who has done this or such this? So in 1953, Waston and Crick produced a double helical model of the DNA molecule. Watson and Crick proposed the model for DNA structure in 1953. Okay, so according to them, DNA structure proposed by Watson and Crick, the DNA molecule is in the form of double helix. It is in the form of double helix. See this form, this is called as the double helix. Here, as per this model, two parallel what threads of nucleotides are there. See first, this one is second. As per this model, two parallel threads of nucleotides are coiled around each other. See, they are coiled around each other, and this arrangement is called as double helix. This arrangement is called as double helix. Here each strand of helix is made up of what? It is made up of nucleotides, and each nucleotide is made up of a phosphoric acid. So we can see this structure can be compared with a coiled and a flexible ladder. Ladder means what? CD. Yes, it can be compared with a coiled and a flexible ladder. Each strand in the molecule of the DNA is made up of small molecules called as known as nucleotide. What it is? Nucleotides, and each nucleotide is made up of what? Phosphoric acid and a deoxyribose what? Deoxyribose sugar and nitrogen bases. So this is the structure of DNA. Now, we have seen that there are four types of nitrogenous bases here. They are of two. First of all, nitrogenous bases are of two types. That is purines and pyrimidines. Purines and pyrimidines. Purines are of two types. Purines are of two types. That is adenine and guanine. Adenine and guanine. And pyrimidines are of two types. That is cytosine and thymine. Nitrogen bases are of two types. Purines, pyrimidines. Purines are of two types, that is adenine and guanine. Pyrimidines means of two types, that is cytosines and thymine. Here in the structure of nucleotide, a molecule of a nitrogenous base and phosphoric acid are each joined to a molecule of a sugar as there are four types of nitrogenous bases. Nucleotides are also of four types. Here see, it is a nitrogenous base, this is a sugar form, and this is a phosphate group. And one more thing which we have to keep in our mind, that adenine always pair with thymine. 80. Adenine always pair with thymine, and cytosine always pair with guanine. Yes, here the helicals have hydrogen bonds with the opposite sides, and the adenine always pair with thymine with a what? With a double hydrogen bond, while cytosine always pairs with guanine with a triple hydrogen bond. Yes, first of all, What? Adenine and thymine, they are pairs. They are always in pair with how many bonds? Double hydrogen bond. But cytosine and guanine, they are always pair with a triple hydrogen bond. Understand up till here? So the deoxyribose sugar and the phosphoric acid alternate with each other and are arranged on the outer side of helix. They are arranged in the outer side of helix. See here, this is a molecular DNA. They are present in a outer side of helix, and the genes in the form of nucleotide sequence are located on the DNA molecule, which is transmitted through parental DNA to the offspring. See here, you can see purine, pyrimidine, GC, TA. You can see here. Yes. See the bonds. In T and A means thymine and adenine, how many hydrogen bonds are there? 2. In guanine and cytosine, how many bonds are there? 3. And see there how they are arranged. They are arranged in a alternative way. Understand up till here? So this is a molecular DNA. We have seen the structure of DNA. Yes or no? Yes. So moving and they are in a anti-parallel position. They are not parallel, they are anti-parallel, means their directions are opposite. Yes, their directions are opposite. So now moving further to the topic, that is genes. Yeah. Now what are genes? Each chromosome is made up of a single DNA molecule. Yes, each chromosome is made up of a single DNA molecule. So segment of this DNA molecule are called as what? Genes. Segments of the DNA molecule are called genes, and due to this variety on the sequence of nucleotides, different kinds of genes are formed. Yeah, due to this only different kinds of genes are formed, and these genes are arranged in a line. These genes are arranged in a line, and these genes control what? These genes control the structure and function of the cells and of the body. One more thing, it also that they transmit the hereditary characteristics from parents to the offsprings also. Yeah, what is the function of the genes? To transmit the hereditary characteristics from the parents to the offsprings, and hence they are said to be the functional units of heredity. For this reason only they are called as a functional units of a heredity. That is why many similarities are seen between parents and their offsprings. So the information about photosynthesis is stored in the genes only. So here you understood what is genes. So genes are defined as the functional units of the heredity. The nucleotide sequence present in DNA molecule is a gene. Thus, genes are seen on the DNA molecule. Genes control. What is the function of the genes? It is to control the structure and the function of the body, and it also helps to transmit the heredity characteristics from parental generation to the offspring. This is all about what? This is all about genes. This is all about the genes. Now, then the next topic is what? Human Genome Project. Now, what is this Human Genome Project? Human Genome Project. Now, in April 2003, researchers announced that the Human Genome Project has completed a high quality sequence of essentially the entire human genome. In April 2003, the researchers announced that the Human Genome Project had completed what? A high quality sequence of essentially the entire human genome. Okay, so in 1990, the Human Genome Project was altogether undertaken by all the genetics of the world. And in June 2000, scientists of this project and Calera Genomics Corporation, that is a private industry in USA, collectively what they have done? Collectively announced the discovery of the complete DNA sequence of Human Genome in 2003. So this sequence closed the gaps from a working draft of genome which was published in 2001. So it also identified the locations of many human genes and provided informations about their structure and organizations. Okay, it also identified the locations of many human genes and provided informations about their structure and organization. So the project made the sequence of the human genome and tools to what? To analyze the data freely available via the internet. This project made the sequence of the human genome and tools to analyze the data freely available via the internet. So in addition to the human genome, the Human Genome Project sequenced the genomes of several other organisms too, including Brewer's yeast, the roundworm, and the fruit fly. Not only here the project was done on the human beings, but also the genomes of several other organisms, including Brewer's yeast, the roundworm, and the fruit fly. The studies were done. So in 2002, the researchers announced that they had also completed a working draft of the mouse genome So by studying the similarities and differences between the human genes and those of other organisms, researchers can discover the functions of particular genes and identify which genes are critical for life. So by studying the similarities and differences between human genes and those of other organisms, researchers can discover the functions of particular genes and identify which genes are critical for life. So this is all about the what Human Genome Project, and it was the seeds of technology. Yeah. Now moving further to the topic, that is DNA fingerprinting. So now what is DNA fingerprinting? This is a new topic which is taken for you, that is DNA fingerprinting. The sequence of the genes in the DNA of a person is called a genome. Yes, the sequence of the genes in the DNA of a person is called a genome. So by DNA fingerprinting technique, a person's genome is identified. Yes or no? By DNA fingerprinting technique, a person's genome is identified, and it is also used to identify the lineage and to identify criminals, because it is unique to every person. As we are knowing that each and every cells are showing different DNA structure. So this DNA fingerprinting helps to identify criminals because it is unique to every person. Understand up till here? So this is all about the DNA fingerprinting. So now I would like that we will see the quick recall of this part which we have seen today. What we have seen today, the topic which we have seen today is DNA. What is the pet name of DNA? It's a master molecule. What is the full form of DNA? It's deoxyribonucleic acid. Yeah, what is the full form of DNA? What is the full form of DNA? Deoxyribonucleic acid. How it looks? It looks just like this. This is the double helix structure of this DNA. This is a double helix structure. Then who has find this? So the scientist, Swiss biochemist Friedrich Meiker in 1869, while studying these white blood cells, he has seen the nucleus. And what he have done? He has collected different pulses from the bandages at a local hospital, and then he added what? A weak alkaline solution. And what he has seen? That he has seen that the nucleus are moving out of the solution, and from here he understands that he isolated a nucleon, and later on he renamed it as DNA. Okay, he was the person who has discovered and DNA. So this DNA, it is not only present in the bacteria, it is present in virus, it is present in plants, it is present in animals, it also present in human beings. In each and every cells of the living organisms, this DNA is present. Okay. Now, where this DNA is located? The DNA is located in each and every cell of the body, and in cell there is a presence of nucleus, and in the nucleus there is present a chromosome, and in chromosome this DNA is located. In chromosome this DNA is located. Okay. So now, but as in our previous slide, what we have seen that these chromosomes are in the double helix form. But when this uncoiling of chromosomes is done, what we see when these chromosomes are uncoiled, see here we can see the supercoils. Yes, and these supercoils are having what? Coils, then nucleosomes, and in the nucleosomes these red balls are called as histones, which are for the responsible for this DNA structure. Okay, this is all about the chromosomes. So what is DNA? DNA is a deoxyribonucleic acid. Then chromosomes are mainly made up of DNA and proteins. It was discovered by whom? By a Swiss biochemist Friedrich Mieke in 1869 while studying the white blood cells. Present in where? Present in nucleus, so called as nucleic acid. It is present in all organisms from viruses and bacteria to human beings. It also controls the functioning, growth and division, that is reproduction of the cell, and are therefore called as what? Master molecules. What they are called as? A master molecules. So what is the structure of DNA? Who has discovered the structure of DNA? In 1953, Waston and Crick produced a double helical model of the DNA molecule. Who was it? Waston and Crick. Now, here as per this model, two parallel threads we can see here, nucleotide are coiled around each other, and this arrangement is called as what? Double helix. Just now I have explained you. And they are compared with what? Coiled and flexible ladder. Yes, and each strand in the molecule of DNA is made up of what? Small molecules, that is nucleotides. It is made up of nucleotides. So here there are four types of nitrogenous bases, that is adenine, guanine, cytosine and thymine. Adenine and guanine is called as what? Purines, while cytosine and thymine are called as pyrimidines. In the structure of nucleotide, a molecule of a nitrogenous base and a phosphoric acid are each joined to a molecule of sugar. As there are four types of nitrogenous bases, nucleotides are also of four types. Nucleotides are of also four types. Here, adenine always pairs with a thymine and cytosine always pair with guanine. See here, you can see the phosphate group, the sugar group and the nitrogenous bases. Understand up till here? We have seen this topics. Then we have seen the molecular form of DNA also. See here, you can see thymine is bonded with a adenine with a two hydrogen bond. Guanine is bonded with or paired with the cytosine with the three bonds. Yeah, three hydrogen bonds, and they are in the anti-parallel directions. This is a molecular DNA. Then what we have seen, the project. Which project? Human Genome Project. So what was the Human Genome Project? Human Genome Project was, what was the Human Genome Project? So Human Genome Project is a great work in the field of human genetics, which has been done by few nations. And according to the findings of HGP, there are 20,000 to 30,000 genes in the human body. And the detailed finding of these reports are available on the website. So later on, scientists discovered the genomic sequences of many microorganisms, and due to the research in genomics, disease causing genes can be identified. And if disease causing genes are identified, genetic diseases can be diagnosed and properly treated. So this project was very useful for us, for human beings. Yeah. So it is where the genes, different genes were studied. So this project was very helpful for us, that is Human Genome Project. One more topic which we have seen, that is DNA fingerprinting. This topic is new for us. DNA fingerprinting, printing. The So what it is, the sequence of the genes in the DNA of a person is called as what? Genome. And by this DNA fingerprinting technique, a person's genome is identified. A person's genome is identified, and what will happen? It is also used to identify the lineage, yeah, lineage, and to identify criminals, because it is unique to every person. So this is all about the DNA. So in a further part, or in a further portion, or in our further lecture, we will study about the ribonucleic acid, that is RNA. So today we have concluded our two parts of this chapter. So I am very thankful to my students that you have heard my lecture very carefully and listened it and try to understand it. Yes, Almas ma'am. So, my dear students, I want that you should read the chapter thoroughly, as this is a new topic for you all. So, I want that, and the many new terms are coming in this chapters. For this, it is needed that you should read the chapter very cleanly, okay, so that you can understand each and every term of this, and it will be useful for you. And this chapter is very important as a exam point of view, okay. So I want that you should read, learn the different short notes, definitions, the structures. You have to practice the diagrams also here, okay, so that it will be helpful for you to attempt the questions in the exams, okay. So in our further part, what we will see about the ribonucleic acid, yeah, that is tomorrow only, and the Mendel's principles of heredity. So I want that you should read the lessons, yeah, so that you can understand. Yes, Almas ma'am.